So thank you, Ethan. I can say that this is not our niche, as you will see from all our data, that we're really happy to share the niche. And I have to say this is also a really awe-inspiring auditorium and also really tremendously amazing projection screen, because it's definitely the largest hair graft I've ever seen. So what I thought I'll do the next half hour or so is to share with you some of our efforts where we try to understand how specialized mesenchymal cells in the hair follicle called dermal papilla cells and the embryonic precursors during embryonic skin development interact with and instruct stem cells and progenitors that leads to and controls hair follicle formation, growth, and regeneration. I'll also make the case that despite many years of research, we still have a rather rudimentary understanding of what the molecular players are and how these cells actually accomplish this feature. And I will try to convince you that it really takes a systematic approach where we look at entire transcriptome to understand these cells in the first place and share all this transcriptome data with the community so that the hair research community can get together in a concerted effort in the next few years to really tackle how these cells work. So this cartoon shows the three key stages in the life of hair follicles, where we believe that these dermal papilla cells instruct neighboring progenitors in the matrix that controls matrix proliferation, migration upwards, and differentiation into the hair shaft. Likewise, during the adult hair cycle, DP cells are thought to activate again stem cells in the germ and in the bulge that leads to new downgrowth and formation of the next hair follicle. Preceding all of this, precursors of the mature dermal papilla cells, so-called in this cluster, so-called dermal condensates, interact with placoid progenitors that leads to hair follicle formation in the first place. And so we're very interested in figuring out what these inductive signals are in the dermal condensates in the DP. And we're very interested in learning what really controls this inductive and instructive fate in the first place that distinguishes these cells from specialized cells from regular fibroblasts. And so this is not only very interesting stem cell niche model where neighboring cells instruct stem cells and progenitors, which is a biologist's dream to figure out these mesenchymal epithelial interactions. It also has practical implications for translational purposes, because if we would understand these inductive signals, or would be able to propagate hair inductive cells, competent cells that can induce hair follicles, we could possibly treat baldness, like here in this cover of The Economist, or perform better skin replacements, including hair grafts, like here in this transplant on the scalp. And so if we would figure this out, we could possibly open shop sometime, like for instance this hair center here in Harlem, in Manhattan, close to where I live, that seems to be permanently closed. So what I thought I'll do today is, in a few slides, I'll give you a brief historical perspective to put everyone on the same page with respect to the discoveries about hair inductive properties of the DP, show you some recent insights into the transcriptomes and some molecular players within the DP, and then show you with some unpublished data in the end what we think will be next and what we possibly could do in the near future. So it really goes back to pioneering work from Colin Yahoda when he was with Roy Oliver now over 30 years ago, who transplanted pellets of cultured rat whiskered DP cells into amputated DPs and showed that this could induce growth. Similarly, 10 years later, together with Amanda Reynolds, he showed that these cultured DP cells, when sandwiched into a hairless footpad, could reprogram or transdifferentiate these epidermal cells to induce follicles. Around the same time, a more quantitative assay was developed by Ulrike Lichte and Wendy Weinberg, who showed rat whiskered DPs, cultured cells, could induce follicles in this chamber graft assay. Again, 10 years later, when I was a postdoc in Elaine Fuchs's lab, we developed now with genetic tools to isolate pure DP cells and show for the first time en masse with en masse isolated cells from the back skin of mice that together with keratinocytes in this chamber graft assay, they are really the cells that can induce hair follicle formation. And so one problem in the field is really that when these DP cells are isolated and propagated in culture, both in mouse and human, and many companies have tried, that these cells still lose the hair-inducing activity. Several researchers over the years tried to figure out signaling pathways that could possibly alter this phenomenon and propagate the hair induction activity. Bruce Morgan has shown over 15 years ago that Wnt signaling plays an important role, yet hair-inducing activity is lost. We have shown later that BMP signaling also is beneficial, but yet hair induction is lost. Claire Higgins, when she was with Angela Cristiano, recently showed that aggregation, that the cells like to aggregate, aggregation has also beneficial effects. So how do these cells get set up in the first place? And so to understand how DPs function, it may really make sense to understand how the earliest precursors really function. And so I will also make a case in this timeline to show you just very few examples and how little we know about how hair follicle formation takes place. And so we believe from early recombination experiments since the 70s that the dermis really instructs the epithelium to form the appendage. We only know since a few years that Wnt beta-catenin signaling plays an important role, yet we still do not know what this patterning signal is that leads then to placoid formation. We know that FGF20 plays a role now in signaling back. We do not know the details of FGF20 signaling within the dermis. We do know that this is instrumental for dermal condensate formation. So then we believe that signals between dermal condensates and placoid then leads to downgrowth in hair germ and hair peg that ultimately leads to the formation of the hair follicles. Yet at this stage, we absolutely have no idea what the signals are that are derived from the dermal condensates. And so we have taken a large-scale approach in the lab to tease out the genes and hopefully then in the future the signals from the dermal condensates. So we isolate both the dermal condensate niche and the placoid progenitors and define molecular signatures by isolating pure cells and interrogating the transcriptomes with next-generation RNA sequencing. And so this was really work spearheaded by Rachel Sennett, a former MD PhD student who graduated last year, who set up a system, quite sophisticated system, to isolate the dermal condensates, also the neighboring fibroblasts for comparisons, the placoid progenitors, the neighboring epidermal keratinocytes, but also the neurocrest-derived melanocytes and the Schwann cells, everything at the earliest stage of the first wave of hair follicle formation in the mouse back skin. And so she did this by setting up a multicolor labeling system where we crossed SOX2 GFP mice and LEF1IFP mice, where we heard earlier already that SOX2 is expressed in the DP cells and it's also expressed in the earliest dermal condensates, while LEF1IFP is expressed at this stage in all fibroblasts, including the dermal condensates. We also learned that melanocytes are RFP positive. Then in combination with two cell surface markers, E-cadherin and P-cadherin, we could isolate placoid-enriched population and also all the other epithelial cells. And so this nested FACS plot just shows you how this beautifully worked. So every dot represents a cell, and you can see here that we get on all the live cells. We can tease out the placoid progenitors separately from all other epithelial cells, shift over the melanocytes separately from the epidermal cells, and from the dermal gate we can sort out pure dermal condensates, fibroblasts, Schwann cells, and two mixtures. A big point that I would like to make here is that here this negative gate contains all undiscovered cells. It contains smooth muscle cells and endothelial cells and any others, and immune cells. An important point here is that this is a representation of the anti-embryonic skin. And so that comes in very handy because we can now look at a few markers that we have previously known, like DKK4 shown in the placoid, highly enriched, not present anywhere else, showing how pure these populations are. The same is true for several other markers. For instance, TVX18 in the dermal condensate, I will show in a few moments how we discovered and how we use this gene. So then we performed next-generation RNA sequencing, and this was collaboration with Amélie Reza in the lab, and this single line here was really work of about 1 year to figure out how to get really deep transcriptome coverage, starting with very small amounts of RNA and coming from low numbers of sorted cells. And so we collaborated then with Avi Mayan and Si-Chen Wang to do a lot of global analysis, and I just show you here the hierarchical clustering where you can see across the different populations the differences in the transcriptomes, and that allowed us then with bioinformatics to tease out signatures of highly enriched genes. So we have now information of a few hundred genes that are highly enriched in dermal condensates, same for fibroblasts on the other end of the spectrum, placoid progenitors, or regular epidermal cells. So first thing we did is we looked into all the genes that we knew from the literature, and it was really exciting to see that all the published genes, virtually all the published genes, are really highly enriched in each of the signatures, and just show you here the dermal condensate and the placoid progenitors. But then the real strength is now that we have many new signature genes, including many members of signaling pathways, many transcription factors, both in the placoids and in the dermal condensates, suggesting that these genes may play a role in the signaling interaction and in setting up the fate of these cells. And so we have now this information, and all these genes are up for grabs, and hopefully this will spark a lot of future studies to tease out the function of placoid progenitors and the niche. And so I just show you here that the validity of all these isolations, just with the QRT PCR, how strongly each of these genes was enriched. And then we could also have a global look at many of the interesting signaling pathways that now came out, because we have now a bird's eye view of all the different genes in so many different cell types. And so what stood out for us is to come up with a hypothesis that axon guidance signaling cues could play a role in cell sorting that takes place. And so in this cartoon here, you can see the dermal populations in the outer ring, the epithelial populations in the inner ring, and we found many genes expressed in all these gene families that play a role in cell sorting, for instance, neurotrophins, semaphorins, and also robos and splits that could play a role in cell sorting that takes place when the condensates form. And so this was the gene discovery to come up with this hypothesis, but now, of course, it takes now a lot of work to tease out the role of these genes. An important point that I made already in the beginning is that it is really essential to share these insights, these transcriptome-wise insights with the community. And so we created this platform, hairgel.net, short for Hair Gene Expression Library, where one can simply look up any gene. And we made this very accessible and also mobile. You can simply look this up on your phone right now, and I would like to ask you to refrain on of my talk, but you could simply test ask for SOX2 and see the dynamic arrangement of the gene expression. So to summarize this first part, I think we have now really very comprehensive information about the earliest the transcriptomes and of the earliest cells during hair follicle formation within the dermal condensates and in the placoids and all the neighboring cell types. We have many new transcription factors and signaling molecules that now need to be followed up, and anybody can now do so by going to Hairgel and really pick the favorite genes for further studies. So, of course, we want now to set up systems to really study the gene function. And so the way we do this is by using reverse genetics and gene ablation, as we have heard already in several talks today. And so to this date, there has not been a single tool where we can target directly the dermal condensates during the earliest formation. And so Laura Grisanti, from a postdoc in the lab, she screened multiple different reporter mice, and we came up with TBX18. TBX18 drives quite specifically early on in embryonic day 14.5 expression in the dermal condensates, and you can see that here. The expression becomes slightly later, a little bit more widespread. We have now also a tamoxifen-inducible version of CRE, and in the meantime, we heard earlier already about SOX2. SOX2 driver for tamoxifen-inducible version of CRE also works quite well. So we have knocked out a whole host of genes over the years, and I just show you the results for two. So Su-Yi Tsai, a former PhD student in the lab, she was very interested in this prominent developmental pathway. We heard already from Fiona today also about Wnt beta-catenin signaling, plays a very important role in hair follicle formation in the placoid. I told you earlier it is very important preceding hair follicle formation broadly in the dermis. We asked the question whether it plays a role directly in the dermal condensate. And so when we crossed the beta-catenin flox mice with the CRE and with a Wnt reporter, Batgal, you can see here in blue the Wnt activation in the condensates and the placoids, and that is gone in the dermal condensates in the knockout. And so in this Holman view, you can see the consequence of this ablation is that the large guard hair follicles do not form. All the other follicles still form because we know with careful analysis that we do not have robust ablation in the other hair types. So to have a deeper look at what could be downstream molecularly of Wnt beta-catenin signaling, we used the genetic trick and crossed these mice now with the SOX2 GFP reporter, could isolate the cells from the knockout and from the control, perform microarrays, and see many interesting genes upregulated and downregulated. including, for instance, FGF10 and inhibin beta A, which is downregulated and has been previously implicated in hair follicle formation, suggesting that these genes could be downstream of Wnt beta-catenin signaling. Another interesting gene, as I've shown you already, SOX2 is expressed in the dermal condensates. It is an important transcription factor in embryonic stem cells, in the neural stem cells, and in the skin it's expressed in Merkel cells, and in the dermal papilla, and in the precursors in the dermal condensates. And so Carlos Clavelle from our postdoc was very interested in knocking out SOX2, and what you can see here is very robust ablation. But to our surprise, we found that SOX2 in the dermal condensates itself is dispensable for hair formation. All the follicles form quite normally. So this was frustrating and very surprising to us, and we surmised there could be functional compensation through genetic redundancy. And I do not have the time to show you, but we have knocked out several other genes that do not show a phenotype, and we really believe either these genes are evolutionary junk, or indeed the niche is a very protected environment. And for SOX2, we're digging deeper, and we're having now ongoing experiments to figure out which SOX could possibly compensate. And so first we need to find out which SOX is really, SOX family members are expressed in the dermal condensates. You can see here phylogenetic tree. There's a whole host of, a whole large family of SOX transcription factors. And we can now simply interrogate hair gel. You can see here a heat map, and you can see 5 different SOXs that are highly enriched in the dermal condensate. And so we could, of course, verify all of this enrichment of these different SOXs, and we have now taken upon us these really long-standing experiments to figure out the what we call the SOX code by crossing all the different imported floxed and gene-targeted mice. And so we are right now in the double stage, and I hope we will be able to report some interesting insights later into the complex regulation by SOXs in the niche. An interesting aspect regarding SOX2 is that, as we heard earlier today, SOX2 is also expressed in the DP of 3 of the 4 major hair follicles in the DP. And so the question simply is then, how can SOX2 possibly regulate genes within the niche that then interact with the matrix progenitors that could regulate either proliferation, migration, or differentiation? So when we had a closer look at the hair shaft outgrowth, we noticed that the hair shaft outgrowth was diminished in the slowed down in the knockout, and this was this length difference was increased over time, suggesting that the machinery of hair shaft production is slowed down. So I do not have time to show you, but we have no increased cell death, we have no changes in proliferation or differentiation, which told us that it could be possibly migration, and I think this is indeed the case. So we performed Ball's chase labeling experiments where proliferating cells take up BRDU, and you can see here normal uptake also in the knockout. When we then chased it out, and I'll show you here the 36-hour time point, we can see that the labeled cells arrive in the differentiating compartment with a delay, and this is here the quantification over time, suggesting that the migration in the knockout progenitors is indeed slowed down when SOX2 transcription factor in the niche is ablated. So to interrogate how this could possibly be regulated through mesenchymal epithelial interaction, we crossed again the mice with the SOX2 GFP reporter and could now isolate with GFP from both control and again from the knockout DP in green the cells, could perform microarrays and found many interesting gene changes. For instance, what we focused our attention on is the BMP signaling pathway, because a BMP6 is strongly upregulated while a BMP inhibitor, SOS-DC1, is strongly downregulated, suggesting that in the knockout scenario there could be de-inhibition of BMP signaling. So we verified that SOS-DC is indeed a target gene of SOX2, and I also show you here that SOS-DC is indeed strongly downregulated in the DP. And to test the outcome of BMP signaling, if this is now increased and indeed altered within in the knockout scenario, we stained for smart phosphorylation, which is downstream of the BMP pathway, and indeed we found precocious and extended expression domains of smart phosphorylation, suggesting that BMP signaling could affect the migration of matrix progenitors as they migrate upwards to differentiate. And so to test next whether BMP signaling could indeed affect migration, we performed an in vitro scratch wound assay. When we see normal migration towards the wound, with BMP6 we could block this, and with SOS-DC we could alleviate this effect. So this allowed us then to arrive at a model where we believe that SOX2 as a transcription factor in the DP regulates many genes, among them SOX1, which fine-tunes the level of BMP signaling, which is altered in the knockout, and it affects migration. I don't have time to show you, but in the shortest hair follicles and the slowest growing hair follicles and the zigzag hair follicles, where SOX2 is not expressed, we have by default increased BMP signaling, suggesting that the migration is naturally slowed down in these. So to summarize this second larger part, I've shown you that over the years we have now developed Cree tools to directly study genes within the dermal condensates. I've shown you that 2 examples, that Wnt beta-catenin signaling is required for guard hair formation, and we believe that the dermal condensate may be a protected environment through functional compensation via genetic redundancy. And I've mentioned before, we have knocked out several genes, and we have published these results in a new category in experimental dermatology for mouse mutants that do not have a phenotype, and I encourage everyone to also send their work there in case you end up with such results. But for SOX2, I told you we have now efforts to figure out which SOXs could possibly compensate. And lastly, I've shown you that in the postnatal DP, SOX2 in the DP controls hair growth through fine-tuning BMP signaling in hair shaft progenitors. So in the last few minutes, I'll just show you some very recently published work just a couple of months ago, and also some unpublished work, how we now branch out towards the postnatal stages during hair, to figure out the transcriptomes of all the important cellular players during hair follicle growth and during the cycle. And so Amélie Reza, a postdoc in the work, a postdoc in the lab, did really this tremendous work to isolate 14 distinct hair follicle and skin cell populations with using 6 different transgenic reporters in 4 combinations plus antibodies to tease out for the first time all the different DPs from different hair subtypes that in the hope to identify genes that could possibly regulate different hair shapes and sizes of the different hair types. She has also isolated for the first time what we call the stem cell precursors as they transition from the plaque stage to the adult bulge stage. And she has isolated for the first time also this subpopulation within the matrix, the transit amplifying signaling pocket. And so Amélie will talk about this work in greater detail on Saturday in the concurrent session, in the hair session. But I only make the point that we now have signatures for all these different cell types, and I forgot to mention that the isolation of the stem cell precursors was a collaboration with Ray Yi from Boulder, Colorado. And so we now can see that the stem cells cluster nicely with the outer root sheath, the transit amplifying cells nicely with the matrix, and all the DPs cluster together. And I will not go into detail, but we believe there are really only they are highly similar and there are only minor differences, and we find several interesting genes, including microRNAs, suggesting that there could be regulation at the post-transcriptional level. And I will not show you many more details except in one slide where we collaborated with Peter Sandstra and Wendy Schauer from University of Toronto, where we now had a closer look at signaling the signaling pathways that are maybe operating between all these different cell types. And so what we did is we used an unbiased approach to map out all the different signature ligands and receptors. And you can see here a collection of all the receptors in color codes, all the different cell types and the ligands and the directionality of the signaling. An important point I would like to make also is that this seems to be highly complex. It is even more complex if we relax this criteria and look for any ligand and receptor that is not necessarily a signature gene. And I will make the case that this will take a whole village to figure out what all these signaling pathways mean in the next few years. But now we have this information, and again we added another tab to hair gel for any researcher out there who is interested in figuring out any of these signaling pathways. So in the last part, I'll show you some of the efforts for, I just mentioned briefly that we're also now trying to solve this during the hair cycle, work that has previously been done by Valentina Greco, who isolated the DP, and also pioneered the isolation of the germ stem cells. We use now her method for the germ stem cells and methods to isolate the bulge stem cells that were pioneered by Rebecca Morris and by Cedric Blampin. But we now have, with triple transgenics, also the possibility to isolate really pure DP cells and all the other neighboring cells, including all the other epithelial cell types, pure fibroblasts. But the important feature here is that we will gain very high sensitivity of gene discovery because we isolate all these 7 populations 4 times in 4 different zones from a single mouse to what we write the wave of hair regeneration, because we know during the first cycle the hair regeneration wave starts in the head region while the tail region is still in the collagen destruction phase. And so in this way we have isolated now 28 populations, and we have now 4 billion reads, and we are sifting through this data and trying to make sense of them, and also will share that with the community. I have only one more data slide where I will make the point that for many years we have the possibility to study genes directly in the stem cells, and direct studies of genes within the DP has been impossible. So we took this upon us to figure out a driver for CRE within the DP, and so we used CRISPR-mediated genome editing, which is really a true revolution in biotechnology. And so we knocked in CRE-ER in one of the highest expressed genes within the DP, and I think we solved now this problem and have now an inducible targeting of the adult DPs with 4 doses of tamoxifen injection. We can now get 100% targeting in close to 90% of the DPs. And so we're very excited about this, and we're going to publish this hopefully soon, but we'll be happy to share these mice on a collaborative basis. So what I've shown in the very last part is that we just finished the characterizations of the stem cell precursors as they transition to the adult phase, the transit amplifying cells, and all the DP subtypes, and we added this to hair gel. We will do this also for the adult hair regeneration phase during the hair cycle, and that we have now inducible gene targeting also for the adult DP niche that will allow gene ablation, fate mapping, cytotoxic ablation, and also determine and go into detail with the relationship of the recently discovered dermal sheath stem cells that Jeff Bernowski talked about earlier. So with this, I'll end, and I thank again my lab, and I mentioned all the people already. Rachel Sennett is back in med school, and she's an aspiring dermatologist, and also here at the meeting. Sui Tsai started her laboratory this year in Taiwan at the National Institute, and Carlos Clavel started his lab 2 years ago in Singapore at the Institute of Medical Biology. I thank, of course, many collaborators who shared mice with us, and the funding sources without which we couldn't do any of this work. NIAMS especially, and the New York State Stem Cell Initiative and the Dermatology Foundation. And lastly, I would like to advertise that we really could need some help for analyzing all these genes and using these new CRE tools for the adult niche. And so we have a really beautiful campus right next to Central Park in Manhattan, so I would be interested in hiring talented postdocs. So with this, I'll thank all of you for your attention.